This is Brain Ponderings podcast. I'm your host, Mark Matson, and today I have a guest who's going to enlighten us about male-female differences in the brain, how they arise during development, or at least what's known so far, and particularly related to her work, and also how the environment, prenatal, early life, affects the brains of males and females, and there may be some differences there. For example, she's done some work, I think it was either a PhD or postdoc on forever chemicals, or at least bisphenol A, which is one of these things that comes from plastics. And so my guest is Professor Mariha Kundakovic. She's Associate Professor of Biological Sciences at Fordham University up in the Bronx. She's an expert on what's called chromatin, and that has to do with how the DNA, which is very long, my understanding, the length of the DNA in an individual cell is greater than our height of our body. How is that packed into these very minute microscopic nuclei of the cells, and how is that organization influence whether genes are turned on or off? So I'll stop talking and say welcome to Marija.
Thank you. Hi, Mark. Thank you very much for having me here. And it's really an honor to be part of this series. So I'm really happy to also share, everything that I feel very strongly about. So thank you for invitation.
Okay. And you're in an exciting time of your research career. You've got, lab that's well funded and a lot of exciting projects, which we'll hear about in this podcast. But before we get into the science, can you talk about your background? Are you, I know you went to college in Serbia. Are you from Serbia?
Yes. I was born and raised in Serbia, and actually I finished my pharmacy school and my master's in pharmacology in Serbia. And then I came to the United States to do my PhD. And I don't know, I think my education in general, really shaped the way how I do science right now, because I think I started because, I started, my mother was a dentist. So she actually, although I was, when I was, early in life, I very, very early started to do some kind of science competitions in math and biology and chemistry. But, she kind of encouraged us to go to these health related professions, right? So I was in pharmacy school, and I finished pharmacy school, and it was an important, experience for me, because I think it taught me, I would say, at least three important things, right? First of all, that I don't want to be a pharmacist, which I think was important to know. But it also taught me that, first of all, that I love molecular biology. I somehow really discovered molecular biology in my first year of college, which was really strange, because I was like, why I didn't study this, actually. And then the third thing that I think was really important and formative experience is that I really understood very early when I studied pharmacology that I was interested in psychopharmacology, right? Somehow from the very beginning, from college, you have this whole pharmacology, and there are, interesting parts of our body and stuff, but I always was interested in the brain and also in the drugs that affect the brain. So, and I started, my scientific path in the Institute for Biological Research in Belgrade, where I did my master's. So it was very early for me, very, very clear that I want to get into this scientific path. And my first project was on, potential antipsychotics. So, but we were studying these compounds that actually are supposed to have serotonergic or dopaminergic effects, right? And I love that, because we were all there were people that actually were predicting the structure of compounds. Then there were chemists that were synthesizing this compound. And it was really this kind of almost like a drug development kind of a situation. And then we were testing these drugs on, certain serotonin reuptake and the dopamine reuptake, and then, if their effects on certain different receptors. And I loved all of that, but somehow I realized we have all these drugs that are not changing for decades, right? I mean, all these drugs that we are using in schizophrenia, and this was all related to schizophrenia in the beginning, somehow were affecting the same type of receptors, and we think we are gonna do something important about that. But somehow, like every drug that was new was really not doing much more than these previous drugs. And I have to say, because you already mentioned chromatin, but I have the feeling that I have the story that it's really like somehow almost initiate me on that path while I was still in Serbia. I remember I was having this exam in pharmacology, and my one of my professors said, I'm going to ask you hormones. But he said these hormones that were like 200 pages, so, and I remember I took and I was reading and I got into these estrogens. And then, there was this part about estrogens where they were saying in very small font, that, they are acting on chromatin and stuff. And I remember I went to the professor who was a head at that time, and he was a molecular biologist. And she asked me and I asked her about what is chromatin and this, and she's like, oh, this is this new area that is called epigenetics. It just kind of started, right? Yeah, so it really was an interesting situation where I was already kind of initiated into that without really even knowing what that was, right? And then when I went to Chicago to do my PhD, I was really lucky to actually come across this group. This was my mentor, Dennis Greystone, and then there was Erminio Costa, who was the director of the institute, and Sandro Guidotti. These are really pioneers in the field of psychiatric epigenetics. So, and I actually had to switch my programs. I was in pharmacology, then I switched to genetics because I wanted to work with them. And I had to do that so that I could work at this psychiatric institute and stuff. So I feel really very lucky. I think people always ask me, how do you start working on something? I think for the epigenetics part, I think I was lucky. And for the sex difference and hormonal regulation part, I think it was also, drawing a little bit from my personal experience as well. So I think it was a nice combination of the two.
And what in your PhD work in Chicago, what were your major findings?
Mm-hmm. So the major findings of what we were actually working on, and I also want people to understand these were very early steps of this psychiatric epigenetics, right? So there was this idea of this couple of groups, including mine, that the group that I worked with, that maybe epigenetic mechanisms are underlying at least part of the symptomatology that you see in schizophrenia. But now you have to understand that there is brain, and there are these cells in the brain, and there is a chromatin inside of these cells, and this is now somehow changed in relation to many of these different factors that we can say can contribute to schizophrenia, not only genetics, but let's say, I don't know, early life experiences, like some stressful events and stuff, and how this can now change that and lead to something that big as schizophrenia is. So I think that path between this, like how chromatin and DNA is modified to the level where, you see patients in the clinic was always like a very difficult to overcome. It's a huge gap, right? And considering that this was like 2003 when I started my PhD, so we are talking like 20 years ago, the also technology that we had was really pretty limited, right? Because now we have all these genomics techniques and stuff, and at that time, everything was more limited. We could look at a couple of genes. I remember the very first studies in schizophrenia was actually on 10 subjects, right? So now imagine if you come up with someone, to the program officer at NIH and say, oh, I have this study with 10 schizophrenic patients, they would close the door in front of you, because they would be like, we are not entertaining this kind of studies. But this was these were very first steps.
Yeah.
And I think what was really important to actually put this out there. And this happens both with the work of my group, which was, schizophrenia and how epigenetic, factors are influencing schizophrenia development, let's say. I mean, I'm really trying to simplify this, and it's not that simple. And at the other hand, it's something else that we also want to talk about today, which is how these early life experiences are shaping the brain to possibly predispose it to some psychopathology. I have to say, and I like to say this always, when I started my PhD, there were literally 5 papers on neuroepigenetics. And no one believed in this. That was another thing that I think was really important, because you wanted to know a little bit about, chromatin and stuff. I think it's important to actually start with that, because, typically when we think about epigenetic regulation and chromatin, we always think about this early, development, right? Because we all start with this one cell, right? It's called zygote, right? And then the cells start differentiating into different cell types, including differentiated cells such as neurons or some type of glial cells and stuff. So in this whole process, because you're starting with the same DNA, right, you need to start turning genes on and off, right? And the only difference really between cells, such as like a liver cell and some type of a neuron, is going to be which genes are being expressed, right? It's not about the DNA content, it's the same. So, and this becomes really important in the very early stages when you have to kind of make this, different lines. And then, like you are really like having these huge changes in the epigenome, right? And when I say epigenome, I want to actually explain to people what this means, right? Because we have the DNA in our cells, right? And you said already it's like 2 meters. I'm a European, so I will say in meters, but it's basically more than 6 feet long, right? So you need to package it like, really long DNA into this very small nucleus of each cell. And so in order to be able to package, you need this very specialized structure that is called chromatin. And the first level of structure, you have the DNA that is wrapped around these histone proteins, right? And we call these like, nucleosomes. This is the first level of chromatin organization. We also call it kind of a beads on the string. But then you also mentioned that there are 3D, like a higher level of organization when you have this nucleosomes that now are, like kind of getting this some spatial orientation to each other, and you end up having something that you can literally pack into a small nucleus. But at the same time, this is important for packaging, but it is also important for regulating gene expression, right? So this is why we now start talking about development, because you need to open certain regions to start transcribing the genes and close others that are not going to be transcribed, right? So there is this interplay between what we call transcription factors, which are the proteins that need to bind to the DNA, and chromatin, that which has to allow the access of the DNA, because DNA is like, people like to talk about, the library. You can have like a full library of books, right? If you cannot access a certain book, it's like you don't have it, right? And this is the same with chromatin, right? If you close the chromatin in certain DNA area, you are not going to be able to express this gene. So the idea was that, like, during development, you really need to establish these patterns, right? But also the idea was, once you establish the pattern, it has to be stable. Otherwise, how do you still, because it's almost like, your cell knows that cell is a neuron or liver cell because of the genes that are being expressed and because of the chromatin structure that is surrounding certain genes. So this is where I think the whole idea started of like, no, this cannot be happening in the brain, because like, it's like going to be a lot of problems there. And then it was logical, for instance, that cancer can have a lot of these changes. Because we know that cancer cells are, particularly for instance, cancer cell has a lot of this DNA demethylation. And I also maybe it's also important to somehow connect DNA methylation, because I know people oftentimes when they think about epigenetics, they think about DNA methylation, right? So when we talk about chromatin, both DNA and protein histones are part of the chromatin, right? And both DNA and histone can be modified. And this modification, this is typically DNA methylation, but also hydroxymethylation, for instance, are called epigenetic marks or chromatin modifications. Also, histone can be modified in many different ways. Let's say they can be acetylated, methylated, phosphorylated, and so on and so forth. So we have a lot of options, and a lot of these marks can make the difference in terms of how chromatin is being opening or closing. For instance, we know oftentimes if in the promoter region, which is the regulatory region of the gene, there is a lot of DNA methylation, this will recruit the binding of certain modifiers of chromatin that will basically close the chromatin around this area, right? So there's an interplay between the two. So what I want to say, like, the majority of literature that I read when I was, a graduate student was actually cancer literature, because, there was a lot of this work that was done in cancer showing there is this global hypomethylation, which actually would induce a lot of this instability of the genome. Or there are these genes that are very important, like, to protect you from cancer, right? That their promoters become tumor suppressor genes, right? Their promoters become hypermethylated, right? And then they're not expressed, right? So there was this notion of, yes, you could have these epigenetic modifications such as DNA methylation or changes in chromatin and histone modification in disorders. But in disorders such as cancer, right? And in cancer, we've done a lot, like, there are epigenetic drugs that are already being used in clinics, right? But, the difference between cancer and the brain disorders, particularly psychiatric disorders, is that in cancer, you do anything to save the, human life, right? You would kill majority of cells just to save the human life, right? But, brain disorders such as psychiatric disorders are always much more subtle. And this is not something where you are like, okay, so I am expecting to see a lot of changes in gene expression, a lot of changes in the chromatin, which typically is important for development. You would be like, no, that's probably something that is not happening in the brain, right? So I think to go back to my first steps, while these studies were now, when you look back on them, you're like, was really enough to have 10, schizophrenic subjects, or was it really very impactful to do studies in some cancer cell lines? And is this really significant for, brain disorders, when we now know that there are people that make brain organoids and this kind of stuff? But someone had to set up, this new area. And someone has to actually fight for people to, in a way, recognize that this may be really happening in the brain, and that this may be really part of the psychopathology that we are seeing. So now, people talk.
Maria, I had a similar experience early on in my career when we started looking at mechanisms of neuronal death, and I was interested in things like Alzheimer's disease, Parkinson's disease, and the cancer field had identified a number of different genes that are important for cancer, like you meant P53, the BCL2 family members, and so on. But at the time I started working on neurons, not much of it, but turns out it's
The
same
same,
mechanisms. It's just different goal. We want to kill cancer cells. We want to keep neurons alive. But we developed actually, there's this one lab that published this low molecular weight P53 inhibitor. And we found and they were studying cancer stuff. And we found it was neuroprotective in some animal models. It never got to the clinic, but anyway, it's the kind of.
It is true. And I have to say, like, you look at this, and they were like also saying something like, oh, there are these, DNMT1 is a DNA methyltransferase that is really important for the maintenance of DNA methylation after each cell division. So how now you kind of think even of why would DNMT1 even have any role in post-mitotic cells such as neurons? So I think this is also like where, there were like these first really, people were very suspicious about these ideas in general. And I really want people to understand that, like, now what's been consider something normal, like, something like, oh, there is a lot of dynamics in the epigenome, like, the cells are changing DNA methylation all the time. That was something that people were thinking, this cannot be happening. Otherwise, you are really end up having some really huge problems, such as cancer or something like that. So what really became for me very, very interesting as we are talking about this shift from development to, and we know it's in neurons, we know this is like, for instance, BDNF, brain-derived neurotrophic factor. It's really something that is very important during development, and it's something that is really important for synaptic plasticity, right?
Yep.
So it does seem that neurons are using similar mechanisms, like, to differentiate into what they are supposed to be, and also to respond to the ever-changing environment. So actually what turned out, and now we know this, but again, it was very difficult to actually accept this 20 years ago, is that the epigenome of neurons is very dynamic, and it's also very, it's very responsive to the environment. And it's not really surprising when you think about it, because you really need something to respond. Of course, people are thinking, oh, they are immediate early genes, but how are the immediate early genes being regulated, right? Or how do they regulate some downstream targets? So it was logical in a way to expect, but I think there was this almost like a fear of, like, how could you change something that was so important to set up the cellular identity into something that is now very dynamic and all of a sudden is involved in plasticity, right? In neuronal plasticity, right? So I think for me, it was super exciting. I didn't really, even you're starting, you don't really understand how important it is to be the part of some new area. But, now going back, like, I feel like, it's been 20 years and I know the first papers. I can tell you who published the first paper of, like, that BDNF gene can be, dynamically regulating in response to neuronal activity. I know who published very first studies on psychiatric changes in psychiatric disorders, and just seeing how that whole field developed over the 20 years for me is absolutely fascinating. Honestly, like, I think you have to be lucky to have such a short life. and still to be able to like, see the development of the entire field. So
So
I
on
think
your PhD work, then you learned a lot of methods to evaluate epigenetic changes, looking at
exactly. So
methylation, and then you manipulated methylation using certain
drugs,
approaches, and then you focused on
two
my
genes.
understanding, a few genes of interest,
Yes.
like there's this gene that's important for production of the inhibitory neurotransmitter GABA.
Uh-huh. Yes.
Yeah,
Get one.
67, and then this fascinating gene relin, which
Really needs another one. It
yeah,
really needs basically another BDNF, right? I mean, because it's at the same time it's important for development, and also at the same time it's
yeah,
important for synaptic plasticity, right?
yeah,
And this is my point that I was also trying to say. I was studying the epigenetic regulation of only two genes. Now, from this perspective, and we do everything like a genome wide, it almost looks like, what were you thinking? Like, you know what I mean? It's like, but these were the early steps, and like, there was like really a lot of supporting evidence that we were actually able to that the reelin and get 67 were actually downregulated in these brains of schizophrenia patients. So it was really important genes to start studying their regulation, both in terms of disease, which I wasn't studying in that time, post-mortem brain tissue. Like, I was just working on this.
yeah.
It was basically neuronal cell line that actually we try to understand. Like, if you manipulate, like, like you add some kind of HDAC inhibitor, which is histone deacetylase inhibitor, which are a drug that should inhibit histone deacetylated and increase histone acetylation, or you inhibit DNA methyltransferase inhibitors, what really happens in terms of changes in the expression. And I found it fascinating. You give this drug and you see induction of these two genes absolutely with the same timing. Like it was very obvious that they were controlled in parallel, like, and like it was very nice to actually be able to do that in the cell culture, where you could actually study the binding to the promoters and of all these regulator proteins, because it was very that time very difficult to study this in postmortem human brain tissue, and still is, right? And as you know, like one of the problems that we have in psychiatric research is like, no study that you do is actually good enough or sufficient, right? Because like, when you study postmortem human brains, you're always like, oh, okay, this is like a dead tissue. These people actually already had the disorder for so long. Maybe something is the effect of drugs, something, like something is the effect of the disorder. So you can't really understand everything, right? Then you have mouse model is like, how do mouse actually develop schizophrenia? You have to study certain
Yeah.
endophenotypes and stuff. It's really difficult, right? Then you have these cellular models that are always like, limited in like whether we are talking about some neuronal cell lines or you're even talking about brain organoids. Once you remove the context of the brain and you can't study behaviors and stuff, like, this is all also very limited. So what I really liked during my PhD is that I was a part of this group that actually studied all these different aspects, right? So there was like they were studying. There was in my lab, someone was studying like postmortem brain tissue. Then there were like Guidotti group that was studying like this in mice. They were having this mouse model of schizophrenia by giving them some kind of methyl donors, and then I was studying this for instance in vitro. So it was very good introduction for me to understand different level. And I now actually tend as a PI to think about through different levels of how we can actually study something. As I hope we can talk about this more in a moment when
Yeah.
I talk about my research.
And then you got interested in brain sex. So males and females' brains are not identical. There's even studies showing that just measuring by MRI the size of different brain regions, there's some differences between males and females. And of course, most people know that females' ovaries produce estrogen, male testes produce testosterone. And so at some point then, I guess in your postdoc work at Columbia, you started to work on brain sex different, so then the question becomes, so I guess two 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 siblings can have pretty much the same DNA. One's a male, one's a female. And, you know, there's a lot of differences between males and females in terms of behavior and so on. So, um, when did you get interested in sex differences, and how did you go about studying what control, like the molecular mechanisms that control brain, I guess call it sexual dimorphism?
Yeah, so I think here, there are a lot of questions. I'm going to try to address this from different angles because I think it's also important. So first of all, I think my interest in sex differences was first of all when I had my own experience too, and I had the feeling that we were not thinking enough about, in general, the effect of hormones on mental health, right? We basically, we're just assuming that, women and men will take the same therapies, that, everything functions the same way. And
Yeah.
that if you see a psychiatrist, the psychiatrist should not take into account sex or gender. I also want to say, and I think it's really important to set this up at the very beginning of starting this conversation, is that when we talk about sexual dimorphism, females, males, we always think about sex and gender as something binary. And this is like where we will going to discuss now, because this is the data that we have. But I want to say, the gender landscape is very, complicated. There are people across the gender spectrum. We also know there are intersex people. So we have to really start to think about how do we also translate this knowledge to people across genders and stuff, because it's really not that everyone, identifies as a male or female or man or woman, right?
Well, I think that's the important point, that these are ultimately behavioral.
Well,
They're behavioral differences,
yeah.
and everybody has different kind of behavioral
Yeah,
tendencies, I guess.
yeah. But I'm gonna try to address this. I just wanted to open up because I
Okay.
feel like sometimes people, if they hear me just say women and men, but people can get
Yeah,
upset
yeah, yeah.
about this for a reason, right? For a reason,
Yeah.
and I'm gonna try to address this by the end. But when I was starting thinking about this, I was really learning about this. There are huge sex differences in the brain, in terms of psychiatric disorders in particular, right? When we talk about sex differences in particular, yeah, you mentioned differences in sizes. The biggest difference is in the brain size, right? The males have on average, a bigger brains than females, but that doesn't mean anything for the function. Let's put it this way. There are multiple studies that are showing changes in the size, in the activity in certain brain areas. I think in the rodents, we know more about certain sexually dimorphic regions. I also want to define that, because I think when you say sexual dimorphism, you really think that there are a very huge quantitative differences, right? In terms of, or qualitative differences, where, something will serve certain function in one sex and certain function in other sex. And I think the idea of sexual dimorphism started from reproductive related behaviors, while I think as the field moved, it became really much more, clear that not only reproductive behaviors are being sexually dimorphic, but that there are sex differences also in the behavior, such as, emotion regulation, learning, your motivation and this kind of stuff, right? So now I think when we talk about the brain in general, it's really difficult to think about the direct, relationship between structure and behavior, right? I mean, it's not like, I wish you ask me some this kind of question, and I can tell you, oh, this brain area is smaller in males, and this is why males do this and females do that. And it's never really like that, right? We know that there are these cross changes, but we don't really know what purpose they serve, first of all. But for me, honestly, and I have to say, and I'm going to connect this to the brain in a minute, for me, what was really the major driving force is this huge sex difference that exists when we talk about psychiatric disorders. And we know that, for instance, if you think about depression and anxiety disorder, that they are at least 2 times more prevalent in women than in men. We also know that schizophrenia is more severe in men, although maybe the prevalence rate is not as much, but, there is a difference. So men actually suffer of more severe symptoms. And then, men has this earlier onset of disorders. Females actually have another peak after menopause, onset. So there are a lot of these differences that were never taken into account. And I was particularly
autism is more common.
autism
Yeah.
and of course ADHD, like several times higher prevalence in boys than in girls.
Yeah.
Alzheimer also more prevalent in women than in men. They're pretty much, I think, every brain disorder you can think of. You may think, oh, so there are sex differences, and I honestly am almost like I can say shocked that no one really wanted to, or at least like in majority of cases. I mean, I don't want to say that there are always people that were interested in sex difference and they were working on that, but this was never really a mainstream research, right?
Yeah.
And for me, it's fascinating that you have a disorder such as depression, anxiety disorders that are the most frequent, like psychiatric disorders that that 2/3 of the population are women, and that first of all, you are not really accounting for that in the clinical studies or in your clinical practice. And second, that the majority of the animal studies were done in males only. So
Yeah.
to the end of my life, you can try to, talk to me and explain this to me, and I will never be able to understand the logic behind that. Because I feel this is such a missed opportunity, right? I mean, don't talk about because first of all, you are missing to find something that is in 2/3 population, maybe it's predisposing, and that you can find something that is really gonna improve the treatment. But second, think about it. Even like as I'm going to talk about it, I believe it's a hormonal component is big. If you find something that is downstream of hormonal regulation, and you can remove, the endocrine component, you can find something that is going to be useful to people across genders, not only to women, right?
Yeah.
So, and this, I understand people would say something like, oh, but there was a complexity. Females have this estrous cycle, but there is a complexity in the brain. There is a complexity in psychiatric disorders, in its psychopathology. And if we try to simplify things, we are actually missing really important points.
Yeah, and for the animal studies, Maria, as a waste animal, think about it. So for decades and decades, all the studies were just in males.
Yeah.
So then the question is, what happens to the females? They're just euthanized and never used.
Used for reproduction, which I have to say,
Well, yeah,
yeah,
but
yeah.
still, I mean,
Yes, yes. I actually have my mentor, Frances Champagne, was telling me because she was studying when I was at Columbia, I was working with Frances. And so she was actually telling me that, she really wanted, because in the lab, everyone was working in males. And she was like, give me females, because, she really wanted to study, maternal behavior, but also how things are passed from the mother to the daughters. And so it was really like that, and I remember, there was one point of time, because we were always ordering, my lab orders more female than males. We always do also males, but we always have more female because we studied the estrous cycle. And at some point, I called Jackson Laboratories and they were like, oh, you know what? We don't really have enough females of that age. And I was like, what do you mean? I think Jackson Laboratories were so used to just have more males than females, that at some point they really had to change the way they actually, breed these animals so that they can actually now, satisfy the demands, because there are more people that study females, right? So I feel it's really, it's such a missed opportunity. But unfortunately, I also, Mark, have to say, I think it really reflects this male-dominated, male-centric culture that we had. And I think something that hopefully is changing, and people really understand that we really have to take into account the sex differences, not only because now you are interested specifically in the women's health, but because first of all, we need to understand the physiology of the brain. And it has to be complete in order, you need also to understand the female brain, because there are differences. And second, as I said, I think it's such a missed opportunity, because there are some disorders and some risk factors that seem to be more affecting females, and that there is a missed opportunity. So, sometimes I talk to people and I would ask psychiatrists, and I would ask them this very specific question. Can you find to me anything that can increase acutely psychiatric risk more than sudden drop in ovarian hormones? But let's be honest, right? The only thing that is more acute risk or equally important is probably some really extreme stressor, right? That would be another one. But think about postpartum depression, right? You have this 20% of pregnant people actually would develop postpartum depression, 20%. And up to 80% would have some kind of a mood change, right? That is, it's severe enough that they would report that as a change in mood. We have premenstrual dysphoric disorder, right? 5 to 8%. This is again when you have ovarian hormone shift. You have menopausal transition, the period when you have 2 to 5 increased risk, and this is before or after menopausal transition. So, when we take this into account, really for me, there is nothing beyond maybe some really extreme stressor that could increase your psychiatric risk this much. And I don't understand how we came to this point that like people were just thinking this is not something that should be like a mainstream research.
I agree 100%. So Mariha, how do sex differences arise during development in prenatal development?
Mm-hmm. Yeah, so I think this is also for me something really important, because I think if you really want to study sex differences, should really think about sources of sex difference, right? So we have three important sets of factors that actually induce sex differences. One is differences like sex chromosomes, right? We have hormones, gonadal hormones, and then we have environment, right? So the first part is actually these sex chromosomes, right? And we know that in majority of cases, people are either having XX, which typically is encoding for the female organism, and then you have XY that is typically encoding for a male organism. So what really happens during development is that at some point, there is a gene on the Y chromosome that is called SRI. It's called a sex determining region that will lead to the development of testes in males. And this will lead to the secretion of testosterone, right? So now this testosterone in rodents actually gets converted into estrogen and masculinizes the brain, while in humans, this testosterone seems to be acting directly on the brain and masculinizing the brain. So now this masculinization is something like, really setting up the stage for later life, right? And we know that during this early life, like, the ovaries are actually dormant. They do not express anything, right? So your default organism is a female organism, but if you have the expression of SRI, development of testes and testosterone secretion, you will gonna end up as a male organism, right? So what will happen then during puberty, you are then having this initiation, increase in testosterone or, testis, like activation, and then ovaries are also starting to produce this cycling, estrogen and progesterone, hormones from the ovaries. And this is actually induced by this gonadotropin releasing hormone from the hypothalamus. This is how it all starts, right? So, and then throughout life, you have this, throughout life, right? I mean, in women, in humans, right? Until menopause, you have these cyclical changes in hormones that are shaping the brain and behavior throughout this reproductive period. And then you have in males, like, the testosterone that is having like a huge roles in many different ways, not only reproductive related behaviors, right? So another thing that I think is really important to mention that I think people typically don't think about that much is that beyond having this role in this sexual determination, right? That there are still XY, sex chromosome complement in each cell of the male organism, and then XX complement in each cell of the female organism. What we also know is that one of the X chromosome becomes inactivated, which again becomes very interesting for my topic of research, because we know that like there are certain genes that are escaping the X inactivation, because the whole idea was that because females have 2 X chromosomes and males have 1 X chromosome, that you need to silence 1 X chromosome in females in order to have the same, they call it dosage compensation, to have the same expression levels of X chromosome in females and males. First of all, we know that there are some genes that escape inactivation. That's already been known. But my lab is also showing that it seems like there is more than that, that there's an interaction between the ovarian hormones and inactive X chromosome, that it's been more dynamic than what we thought it was.
How many genes are there on the X chromosome, and how many of them are turned on in the brain?
There are huge number of genes. Like, I don't want to say the number, but unlike in the Y chromosome, it's really like one, if not the biggest chromosome. But we know that in terms of percentage that, and I think that's also an important thing to mention, that in mice, 3 to 7% of genes is actually escaping X inactivation, while in humans is actually between 15 and 25%. So just to think evolutionary, why it would be important to increase the number of XXKPs this much, for me, it's already very intriguing. I think there is something about inactive X chromosome that is much more important than what we wanted to think of it, right? And then I also wanted to say, so you have in each cell this situation, and like not only that there are like Y-linked genes that are not important for the testis development, but there are these XXKP in females, and then inactive X chromosome also has a very specific structure, right? Because in order to be inactivated, there is like there are a lot of repressor complexes that need to be kind of recruited to this X chromosome. So it actually forms a very different cellular environment than you have in each cell that is XY. And this is something that hasn't been taken into account, but I think really going forward, like it's something really important to actually mention. And the third thing that I also wanted to mention, like beyond like just having these different sex chromosomes, having different gonadal hormones that are being secreted at different levels throughout life. You also have this interaction with the environment. And as you know, like certain environments can be sex specific or gender specific. We know that, growing up as a girl, as a boy, is going to be very different. And you are going to have a very different environment in terms of how people even interact with you. And this can create these interactions. With your hormonal, status and, your biological makeup. And then we know that males and females can differently interact with the environment that is the same, right? So there are a lot of these options. And sometimes when I talk with people would say something like, oh, but yeah, of course, females have a higher levels of depression because of their status in the society. But, of course, there are social factors that are playing the role. But we have to say that no one is this is not in vacuum, right? I mean, you have the interaction between biology and the stressful situation, let's say. And just having a certain makeup, like a hormonal makeup or genetic makeup or something, it actually can predispose you more for the same type of an environment. So I think really it's a missed opportunity. And I try really with my work to really change this. So it's just to give you an example how I'm thinking, right? So let's talk about depression, right? So people will tell you oftentimes that depression is 2 times more prevalent in women than in men, without really explaining that this is pretty much only during the reproductive period, right? So if you look at the depression rate before puberty, they're very similar, if not higher in boys than in girls. If you look post-menopause, the risk between males and women and men is very similar, right? So really the risk jumps to 2 times higher when the puberty starts, when actually girls start menstruating, when you start seeing this fluctuation in ovarian hormones. Then the risk actually increases every time that you have like this bigger shift, right? Postpartum. Then, during the menopausal transition, when you have, actually during the menopausal transition, you have the most extreme hormonal fluctuations. Right? You have very high, you have very low at the level that are very, in a way, erratic. And I think this is what actually makes the risk to be the most profound. So I think what is important for people to understand is that estrogens and progesterone are very protective for the women's health and the health of people with ovaries in general. When we talk about the brain health, we know that actually menopause will be almost like triggering the risk and increasing the risk for Alzheimer's disease later in life. We know now, of course, in the people that are more predisposed, right? Not in everyone, but there are brain changes that are happening during menopause that seems to actually increase the risk for women for Alzheimer's later in life, right? So we know that, estrogen is important for brain health. We know that it's important for metabolic function, right? Because you see all of these changes in the fat mass and activity levels and all of this, once you reach the, ovarian senescence, right? Yeah. So we know that there is a lot of things for which, ovarian hormones are very protective for the bones, for the heart health, for everything. But I think, and from everything that I ever read and all the thinking that I had, I think the problem here is not high or low estradiol. When we talk about mood disorders, the problem are those fluctuations and those a need for the brain to readjust every time when you have this change in hormones. Yeah, so, and really the contribution of my work was that I was always thinking, you know what? This dynamism of hormonal changes has to be reflected somewhere at the molecular level. And because I was the person that was very interested in chromatin and epigenetics in general, I was thinking probably ovarian hormones can actually open and close the chromatin because that's the way how the chromatin in neurons responds to environment. So whether this is external environment, such as, let's say, stress, or this we are talking about changing the hormonal levels, because, during the, let's say, each ovarian cycle, your brain is exposed to like higher and low levels of estradiol and progesterone. So now, why was this so novel? It was so novel because, like, we knew from the studies in breast cancer cell lines that estrogen can actually change the chromatin. That was one of the first things that people actually learned, right? I mean, that was how actually the whole study of the chromatin started. And I actually didn't mention, but I was actually working in Miguel Beato in Center for Genomic Regulation in Barcelona for the period of a little bit more than a year, where they were actually studying this in cancer cell lines. And this was very helpful to me because actually it introduced me to that world of chromatin regulation by sex hormones, right? And then, like I also had that between going to Columbia University, where I worked with Francis Chopin, I also worked at Mount Sinai for a period of year with Sharam Akbarian, where we studied this this was like a big psych-encode consortium project where we were studying actually epigenomic changes in people with schizophrenia versus, control subjects. So this actually, all the training that I had, which started from cancer cell lines to mice and mouse behaviors that I studied at Columbia, to actually these post-mortem studies again with Sharam Akbarian at Mount Sinai, actually helped me, solidify my understanding of the epigenomics in general and epigenetic regulation and, studying behavior. But then I also got really interested in this dynamism that ovarian hormones actually, how ovarian hormones can change the epigenome. And not to mention that there was like the whole hole in our knowledge of this because no one has ever studied this. And I remember when I started studying this, like, it was actually a very risky project because when I started talking to people and telling them what I wanted to do, because we studied this with physiological estrocycle in mice. So mice have something that is very similar to, like a menstrual cycle in humans, right? It's called the estrocycle. It's actually much shorter. It's 4 to 5 days, but actually allows you to get, it's absolutely the same thing, right? It's just shorter, but the changes in estrogens and progesterone are very similar. The ovulation starts, after the surge in estradiol. So it's really everything. It's a very similar physiological system to humans. So, and I was like, I want to study this in physiological estrocycle. And people were like, oh, this is not how you study it. Like, you have to do ovaroctomy and then you add the estradiol. And I was thinking, no, I don't want to study this because I think if you do the ovaroctomy, there are going to be a lot of changes. Then you get estrogen, you don't know which dose. So you really don't. And then also there's an interplay between estrogens and progesterone, right? Estrogens are not also working in vacuum. They need progesterone in order to actually, there's a lot of interactions happening. So this was my starting hypothesis, and I really wanted to do this since I was in my PhD. I always had this idea, for sure, hormones have to change. And again, I said it was also personal in a way because I was feeling this in like I had these premenstrual symptoms that I was like, I would feel the change, in my mood, in my well-being, like, just like, during that early period when you have these ovarian hormone changes, right? Basically the drop just before menstruation. And so I was thinking this has to be happening because, like, our epigenome is very dynamic. But the problem was that, again, going back to this idea that these are post-mitotic cells. And like now introducing the new dynamic factors such as like hormones would be like, oh my God, what is happening in this neuron? But this is exactly what we found. We found literally that going from one phase to another, which is in mice almost like 24 hours, you have like almost 30% of chromatin being reorganized in ventral hippocampal neurons. And the reason why we wanted to study the ventral hippocampal neurons is because, the hippocampus is important for both learning and memory and for emotion regulation, right? And there were studies a lot from Rene Hahn and other groups that actually showed this functional distinction across this dorsoventral, axis. So I knew that basically if you, that ventral hippocampal neurons, if you optogenetically activate them or you lesion that part, you will change anxiety-related behaviors such as, let's say, elevated plasmase, but you won't change spatial memory. And when we started, we actually first looked at the behavior, and we were able to show that when you have high estradiol levels during physiological estrous cycle, females are behaving the same as males. However, when you see this drop in estrogen, you get into this diestrous phase, which is more similar to the luteal phase in menstruating individuals. What will happen is that you are increased anxiety risk, right? Anxiety indices in this simple anxiety test in mice. So in my mind, it was like, okay, we need the brain region that we know that it's driving those behaviors so that we can now show that basically as hormonal mediate changes, that in these specific neurons, there is a change of chromatin opening and closing that leads to changes in gene expression. We also show changes in dendritic spine density in the same brain area with the cycle. And this is actually your
So the number of synapses goes down when estradiol is low?
Yes, yes. And in my opinion is that this is probably a biological substrates for what we are seeing in the terms of behavior. I want to say also in humans, there are studies that are showing, I don't know if you're aware of this, probably a lot of people are not, that there are changes in gray matter across the menstrual cycle in humans. And that these changes are also following the estradiol and progesterone levels, basically, right? So I think when you think
I do
about...
know that there's changes in gray matter with depression in
Yes,
humans,
yes,
and when a person recovers from the depression,
they are
it goes,
recovering. Yes,
yeah. So you're getting these changes in the synapses and dendrites
exactly.
that are influenced by hormonal state and, well, and behavioral state or a mental state.
So this is my, just to actually maybe also make this closer to understanding of, for the audience as well. Like, if you think about it, right? And a lot of these things were done in male mice, and we can make a connection to depression, right? In male mice, if you stress them out, you lose the dendrites, right?
Right.
Dendritic spines, right? This is what you are losing, right? You decrease the number of dendritic spines. If you give them antidepressant drugs like ketamine or something, like you will increase dendritic spines. So there is this now whole idea that this neuroplasticity actually is important for antidepressant activity. And as you were saying, the same happened in depression. There is a shrinkage of the hippocampus in people with depression. And then you see that with antidepressant treatment, also like, other things that can actually be helpful, like, exercise, that this can actually increase the gray matter and stuff, right? So what my point here is that people typically study this in male animals, and they will be like, okay, so with stress, you decrease dendritic spines. With, like exercise or antidepressant treatment, you increase. In female mice, you get that cycling like as a natural thing, hormones, right? So this is my opinion is that like you have the vulnerability that comes with a drop of estradiol because you're losing these dendritic spines, and that this is your like structural, like substrate. While what we are seeing at the level of chromatin and gene expression is actually the molecular substrate for this. And what was really important for me, we did not show this only with a physiological model. So that was the first thing that we did, right? So we first showed that with the astrocycle, you change chromatin organization, not only at the level of these nucleosomes, but you mentioned 3D structure. So the 3D genome rearranges, right? There is a spatial rearrangement with the change in the hormones. Then we were able to reproduce part of it when we do the overoctomy, and then when we replace this with estradiol. But we did also one more thing. And I think for me, this is now a bioRxiv preprint. So hopefully we are going to have this published soon. But what we were also able to do. So when you get this, because this is all like genome-wide data, right? So you study chromatin in ventral hippocampal neurons, and you see thousands, let's say, of genes or hundreds of genes that change chromatin organization. What you can ask as a question is like, what is the upstream regulator of this? Right? Because you can see which DNA sequence is particularly being enriched, let's say, when there is a high estradiol levels. And we found this EGR1, which is a transcription factor that is really important immediate early gene that is responsive also to stress. And so we were actually able to show that that specific immediate early gene increases with estradiol in terms of its expression, and also its sites open specifically during high estrogenic phase. So my graduate student, David Rock, this was his actually thesis. What he did, he took this gene, EGR1, gene expressing the EGR1. He specifically overexpressed this gene in these ventral hippocampal neurons in mice in vivo, and these mice are now over-optimized, so they don't have any cycle. And he was able to recreate a big part of what we are seeing during the high estrogenic phase, right? And this difference, because we did this overexpression both in females and in males, we showed that the majority of changes that males would respond in terms of molecular changes. also synaptic plasticity, but not behavior, number one. And number two, the majority of changes that we are seeing were very sex-specific. So in my opinion, this is the first sex-specific immediate early gene and chromatin regulator that everyone ever shown. And I find this very important because I think it connects something that I'm very interested in terms of, okay, so this is my biology. This is what can be the molecular mechanism for how these ovarian hormones are increasing the psychiatric risk. But then look at this, like if we study this, we can find the factors that we can manipulate and decrease anxiety indices or something. So that actually gives us a proof of concept that by doing this kind of a research, we can actually find new drug targets and actually be helpful to everyone.
You can set up screens and so on.
Exactly.
I was going to say, Marija, so this EGR1, so other what are called immediate early genes, genes that they're called that, I guess, because they turn on very quickly after synapse is active. And so when a synapse is activated, like glutamatergic synapse, you get calcium influx, and the calcium is a signal for these immediate early genes, I think, including EGR1.
Yes.
And so what happens with activity levels in those same neural circuits?
We know that actually estradiol, like a high estrogen phase, is also associated with increased neuronal activity as well.
Okay.
So that's been shown before. It's not done in my lab. We are not electrophysiology lab, but it's been shown before. So I think this all makes sense. I think it's just estradiol.
What you showed is the EGR1 is a critical mediator.
Exactly.
Of that. Yeah.
And what is really also new here is that not only is a critical mediator, but it's also a chromatin regulator, right? So this actually connects this idea of like there is this immediate early gene, but then this is how they can change these downstream targets. So I do want to say that there were like two different groups that actually Zhang group from UPenn, and then there were like two groups that actually showed that these immediate early gene products can be chromatin openers. However, I feel like that this is the first time, because a lot of these studies were done only in males, let's say, and then you assume that what's been shown in males also is true for female. My Greenberg group at Harvard also showed these others. So these two groups, they actually propose this idea that immediate early genes are like chromatin openers, right? But what we showed for the first time is that this is there are sex specific immediate early genes, and they are sex specific because they respond to ovarian hormone changes. So I think this is for me also, if I want to leave a message here for the audience in a way, the message is also there are these sex differences that we really need to explore, right? And I think this can help us understand the physiology, but also understand, how we go about making more efficient drugs. Because what can happen sometimes with maybe there are some drugs failed just because, there was too much noise, women would not respond, or we don't know about these things, right? No one actually ever looked so hard at, the effect of sex. And I think this has to be one of the things. But I think what is really important here also is a lot of times now, because, NIH and other these agencies actually now, they ask people to include females. And I think this is great. I think including females is really the first step. But I think what really is important is that we have to understand, if you want to understand, let's say you study adult males and females and you see some sex difference, you have only a snapshot of one point in time. But if you really want to understand this mechanistically, I think you really have to think about where is this coming from, right? I mean, is this driven by, let's say, hormonal changes? Because if you start thinking in such a way, it's going to be much easier, right, for you to understand which are receptors that are being responsible for this. Where is the pathway? Because otherwise, it's really blind. Imagine also doing just RNA-seq, right? This genome-wide gene expression analysis, right? So what can happen is you're just oh, I saw these genes being induced in female, but not in male. But where is this coming from? What is your target that you can actually address? And is that something that you can actually target with drugs? I think that's an important thing. Coming back, this is also why my pharmacy background is important, because I do always have this in mind, right? I mean, I think a lot of molecular biologists, they are really interested in all this gene regulation. And that's fantastic. I think I love the invisible world of molecular biology. I think this is something for me, it's super exciting. But I think if you want to be more practical, if you want to be like, okay, but I need to understand how we help people, right? I think we really have to understand where is this coming from in terms of what could I target possibly, which is the receptor, what is the signaling pathway, so I think if you start thinking about the source of sex difference, this can actually make us closer to understanding. Another thing that also I want to mention, we showed, and I published a paper that is called Why the Astrocycle Matters for Neuroscience. I made this dramatic, a title on purpose, because I wanted people to start thinking about it, because we were showing also, if you put together all females and compare them to males in anxiety-related tests, you see no sex difference. So you really need to segregate them based on the astrocycle phase in order to understand that dynamic sex difference. And someone may say, but that's dynamic, it's not important. But it is important, because it's exactly those shifts that are increasing the risk. And if you want to understand it, and again, I want to say, and I will say also this 100 times, not every person that experiences ovarian hormone changes is gonna be at some point affected by anxiety, depression. That's not sufficient. We have to also think about interactions with stress, right? And I think this is something that we also wanted to address. I will try to address it very briefly. So we know, for instance, and I think that's also something that we found at the molecular level. When we look at the chromatin changes across the cycle, there are thousands of changes. But only subset of genes, like sometimes only 10% of genes, change the gene expression. However, when you do the pathway analysis of all the changes, you see the enrichment for brain disorder relevant genes, and particularly for anxiety and serotonergic transmission. So what I think is, in a way, that hormones are setting up certain vulnerability in the female organism, right? So there is a vulnerability in terms of there is a decrease in spines or decrease in brain matter. There are some changes, but I think that you need another hit typically for this to actually now become and to be translated into some kind of a psychopathology. And this is why I think it's really important to start thinking about early life stress, when actually this also some of these changes are actually being set up, right? And then to also think about acute stressors and how they can affect, like a risk for disorders. I think this is something that is important. I also want to mention one of the really interesting a piece of data that we have. And this is that basically, if females are exposed to early life stress, sometimes what we are seeing that later in life, you're losing the effect of the estrocycle. And basically, if there was a protective effect of estradiol, as I told you, during the high estrogenic phase, you literally have less anxiety, or is associated with less addiction when we talk about cocaine or something. You lose this protective effect of estrogen if there was early life stress. So now there are different possibilities. One possibility is that if you do early life stress, maybe there are changes in ovaries, and maybe the ovarian development changes. Maybe there is not ovarian development in terms of, but there might be some changes in the ovaries that will affect how females are cycling later on, and how this can be protective. But then there is also a possibility that, the cycle is normal, but the responsiveness of the brain to the cycle is different, because we have to understand that what really responds to the ovarian hormones is the receptors that are in the brain. Yeah. Right. So if the something changes, then so I think these interactions are really important for thinking about that. We have a recent study in Cell Reports where we literally showed, and this was actually a follow-up to our studies that we did. It's an early life stress study that actually was also based on the findings from humans. So when I look into the human literature, what I actually found is that there are differences in the way how women and men get addicted, right? How the substance use the disorder develops. But in particular for cocaine addiction, that early life stress is much more predisposing factor for women than in men, right? And then we were thinking, let's study two sex-specific factors. One factor is early life stress, and another factor is ovarian hormone changes, and how this interaction can actually lead to these changes in the phenotype that we are seeing. So I think there is so much to be done. I think we just need to think more about possibilities. And I think the setup, the mind setup has to change in a way to understand this. Because, like, if I've heard of people, let's say, saying that they had a premenstrual dysphoric disorder that was not diagnosed for years. Because psychiatrists never ask the patient, when do you see your symptoms, right?
Yeah.
So I think, psychiatrists have to start basically thinking, and the education has to, like, be enriched a little bit. I actually was recently, like, a part of the there was at CUNY, there was like this women's health conference, and I learned that they have this more and more this kind of a reproductive psychiatry, like fellowships and stuff. Like,
Yeah,
I
good,
think
good.
there are, but there should not be like one person that is specializing to this. I think every person should have a little
Yeah,
bit of a curriculum where they can recognize the things, right? If you have a woman that is going through the menopausal transition, and you treat that person the same way as if the person is, like, in their 20s, there is a problem, right? You have to take into account the hormonal milieu or something, and then you start thinking, if you are going through menopausal transition. I became very interested in this because I actually want to do the human study of menopausal transition and molecular changes, because we don't know much about it. And I learned recently, like, that brain banks don't have information about this, so we had to actually develop the whole new set of markers to go back into postmortem brain and find the menopausal status, right? So,
yeah.
so I think what is really important is to understand if there is like this huge risk, like, that is associated with this. Like, like you ask people and they're like, I don't know who to talk to, right? Because there is a gynecologist.
The age of menopause is fairly consistent among women, right? It's like, or not.
Nah, yeah, man. On average, it's 51, but you can still be 46 or 7 and be menopausal. You can be 53 and be postmenopausal.
So,
So
so your point is that in a lot of the brain banks, they don't,
they
well,
don't have the information.
they have the age, but
They
they
have the
don't.
age, yes, and they use that. If people would do the studies, they will use age as a proxy, but this is not really a proxy.
Yeah.
Of course, there is a lot of correlation, but, like, it's not sufficient if you really want to understand the actual. So we've done, it's actually in preparation, this new manuscript that we actually.
But do they have a lot of brain banks also have blood samples, and
Not many,
you can
yes.
measure estradiol in the
Yeah,
blood.
yes. This is what we are actually doing. We have the whole steroid hormone panel in blood, and then we also study this in the brain, because some of the banks do not have blood. So that's also important.
But are you
Also,
measuring estradiol in the blood samples?
yeah, so you can measure estradiol, you can
Yeah.
measure
Okay.
progesterone, but we study like 14 different steroid hormones. We also study FSH and AMH. FSH is this follicular stimulating hormone, which actually, because this is the part of the HPG axis, the hypothalamic pituitary gonadal axis.
Yeah.
So what will typically happen is that once the ovaries actually stop producing or start producing lower levels of estradiol, there is this compensatory mechanism where you start increasing FSH from the pituitary in order to kind of push the ovary. So one of the first signs of menopausal transition is also increased FSH, levels. And then AMH is anti-Müllerian hormone. This is also the hormone that actually is the marker of the ovarian reserve. So there are the studies that actually in living humans, but there is still not like a cutoff point. Like the way how you diagnose the menopause right now is literally 1 year after the last period, right? So I think it's more important to, if you want to understand changes in the brain, it's more important to understand the biologically, what are the biological factors you were saying? What are the estradiol levels at this moment? What are the FSH levels, something? But I think there is also one tricky part, and the tricky part is, as I mentioned previously, that during menopausal transition, you have a lot of fluctuations, and you again have only one measurement, right, at a single point of time. So this is why I think having like a kind of a composite measure is going to be more helpful in terms of understanding what, and also take into account that some people might be taking hormonal replacement therapy, some people might be taking, hormonal contraceptives and stuff. So there is a lot of complexity, but I find this complexity very exciting and very important. And I think there is so much opportunities there that to improve women's health, to improve health of people across gender in general. But I hope that this conversation, thank you really for this invitation, actually sparks some interest, and also that people go and read more and find more interest in what we are doing. I don't know if you heard, but there is a whole new White House initiative on women's health. I just hope that this comes also, this money goes into funds, goes on also into NIH, and that, like, there are good calls for the new studies so that we really can help fill the gap in terms of both female brain and women's
Yeah,
health in general. I think it's going to be really
that's
important.
great. Before you leave, so you talked about early life stress, and I had Charles Nelson on an earlier
Mm-hmm.
podcast who's done nice work in these humans and institutionalized kids and so on. But um, there's there's been recently an increase in incidence of mental illness, depression, and anxiety disorders in younger people.
Mm-hmm.
And so, what's the explanation? Some people are saying, "Oh, it's the internet or something." But one thing that's happened for the last 40 years is people are exercising less and they're eating more.
Mm-hmm. Yeah, I
You
think
know,
all. Yeah.
and so those are not good for your brain, and maybe also in case of terms of risk for disorders, but also prenatal, right? I had a podcast on autism, and we talked a little bit about the in utero environment and the idea that the maternal status, particularly obesity or type 2 diabetes, uh, is a risk factor for autism in the babies born. Now, you did some work on, we've also heard a lot about these forever chemicals, and, everybody, when we go to the supermarket, a lot of the food we get's in containers, soft drinks are in plastic often, and so on. And you did some work in at Columbia, right? With
Yes, yes,
Francis
with Columbia
Champagne
was
on,
with Francis Champagne
yeah,
was on bisphenol A.
yeah,
That was my introduction to these early life stress studies. Yeah, so because there was, like, we have all these exposure, these are so-called endocrine disruptors, and again, this is in a way a very good connection between early life, environment and sex differences. Because if you have some like a compound such as bisphenol A, which is very estrogenic compound. It actually affects the estrogen receptors, and it has all these endocrine, effects. Because it's typical. Let's say that it induces like precocious puberty, and,
yeah,
like, there are a lot of reproductive related outcomes that people have shown that change with the bisphenol A. So it's been shown, like, for instance, in humans, that you can connect these increased levels of bisphenol A exposure to, like early life, some changes in behaviors that are externalizing or internalizing. There are sex differences and stuff. So it's not really easy, as you know now, to just kind of connect prenatal bisphenol treatment with the increased depression risk or something like that. That I still don't think we have enough information for something like that. But I think this is just one of those examples and the contributors where I think the whole exposure of the mother, right? So because a lot of this is just particular endocrine disruptor that can go through the placenta, right? A lot of this, like compounds are gonna, change the brain in certain ways. And as you were saying, you probably call them forever chemicals because you can't really remove them, right? I mean, you can now have the bisphenol A free plastics and stuff, but they're all over still, I think, as you said, like, it's a lot of, like food that is being eaten that is not very healthy, a lot of pollution as well. Like, there
yeah,
were studies, like when I was at Columbia, that was also a very important experience with me. We actually had these studies, like with the Columbia School of Public Health, Julie Herbstman and Ricky Pereira. They were actually running these human studies on, like, how, the effect of pollution and these endocrine disruptors on the human brain, right? So we could actually connect what we were studying in mice with what they were studying in humans, right? So we know, like, these are the things that are changing the brain. And so you might actually have, like a multiple of this. There's some genetic predisposition, and then you have, a lot of these compounds that are affecting the brain development. And then, of course, when we start talking about young people, like, I think you can talk about the internet, but I think in general, there's less social interaction, right? As we know, like, we played on the street a lot, when I
yeah,
was a kid. This is this very healthy development of, and now what you have, like, you go, and then it's a lot of, believe me, I think there is a lot of stress just being all the time on this social media. Like,
yeah,
everyone can just snap picture of you post it somewhere online, particularly when we think about puberty, like kids are very sensitive, girls are very sensitive in particular, so you increase this pressure of someone performing in everyday life all the time, right? And this is this is where now I wanted to connect to biology, because I just said girls are very sensitive to this. Yes, exactly, because there is a biology that is different now, and people like girls and other people with ovaries, like it's going to be more sensitive because of the biological makeup. So you will kind of have this interaction between your personality that develop based on a biological makeup and the environment that you're exposed to all the time. So it is stressful to grow up like this, that like you are a little celebrity all the time. You know what I mean? So I feel there is a lot of reasons, not to mention the effects of COVID, pandemic and everything. There is so much social isolation, and I've noticed this with my students. I think they are coming back now, but once we moved from this period of when there was like this a lot of when we were all online to the period when the in-person classes started, I could see that the kids were just really, I don't want to say kids, these are the young adults, like they're young people like in their early 20s. You could see that they almost had to relearn how to connect with people in person, and also like how to take the exams and all of these things. So it's a lot of it's a lot of stress that we are experiencing right now. And I heard actually someone was telling the story about their grandmother, like that was actually living through this like one of the world wars, right? And then they said like that was probably very stressful, and that's the grandmother said like yeah, but I think your life is more stressful now than it was for me going through the war, like what I mean. So I think it's the expectations are all over the place. We work too much, so it's overall the environment actually is set up, like I think our nature is polluted, our nature has a lot of these chemicals. So I think all of this like contributes to this. But then what do we know? Like I think also probably in the past a lot of this was underdiagnosed, right? I mean, a lot of these things would just go as we are now talking about, like who knew about this increase in menopausal risk? Like who talked about that before, right? Almost no one, right? You had this postpartum depression forever, but no one really talked about this much because women just did not feel like they should be complaining in the society that is so male dominated, that if they show a certain weakness, this can be used against them, right? So I'm hopeful that we also are changing our society in a way that people feel more that there is not such a stigma around the psychiatric disorder, so that people feel more free to share their experiences, so that then we can help them, right? So if you don't know about these things, then it's also like more difficult. So I think it's the combination of like underreporting to getting into the environment that becomes very stressful. But biology is the key. This is what I'm saying. Like we have to, of course, think about the ways of how we remove the stressor. This is something that I, as a biologist, cannot do. You ask politicians and people that are more involved in policies how they actually make this society healthier and less stressful. And for me, the question is like, how do I use my knowledge? How our biology actually contributes to risk or resilience, how this really interacts with the outside environment, so that we can actually not only, do some studies that are actually going to be published in these high impact journals and stuff, but we help actual people with psychiatric disorders.
Absolutely. And disseminating this information to the public is important. We still have a problem in this country with kind of very low levels of science literacy.
Yeah.
A lot of people, they may have one or two science classes in high school, and that's about it. And then, but it's kind of rudimentary, their knowledge, including kind of basic things. And it's a big problem because all the people that are in politics and in government, elected government positions, they don't have a background in science.
Sure. Yeah.
And, how do you make informed policies when you don't have, we see this now with internet issues and the artificial intelligence thing, and none of the people in Congress know anything about it, so they don't know how to regulate it. And Yeah. So anyway, education is kind of the key. And
And also like translating this knowledge. This is why I said, like, for me, it was really important to talk about, to have this connection with these people at the School of Public Health, because they literally change policies. Their work changed
yeah,
the actual policies, right? So they actually contributed to this idea. I was actually it was really weird in a way, very strange to be part of this BPA project, because this was the only time in my life where I thought I was also almost like, like followed, by these people that are policymakers. Like, there was like literally a document in some European Commission where they cited our PNAS paper on bisphenol A like 30 times. Right. And that was a little bit scary, because then you realize, yeah, I'm really doing this impactful work, but also like, some people are losing a lot of money because of this. And then, this can be like, an important, you can be like not target in a certain way, but like, it's a lot of
well,
responsibility.
and the
And to
plastic companies, they they have lobby people in
exactly. Yes. Yes.
Washington too, right? And
For sure. For sure.
they're trying to minimize
The
that.
actual scientific output. So this is actually the first time that happened to me, that someone tried to almost criticize our statistics, how we came up with this. So I was just like, really, like, but then at the same time you also feel very proud, like, that your work is actually being used, and it's truly like applicable right away. So
Yeah.
that's what I'm saying. I think it's really important also as a biologist to have this connection with people that are both clinicians and also like people from public health that can really make the difference also where we need it, right? Not only in terms of course, the generating new knowledge is always beautiful, right? And this is something that why I'm here for primarily. But then again, like we want to be able to translate this knowledge to both clinical practice and also to the policies.
Yeah. Yeah. Great. Thanks for taking the time. I'll let you get
Sure.
back to your lab. I know you've got a lot of eager, young budding scientists there who are,
True.
yeah. So,
Yeah. Thank you
okay.
so much, Mark. It was really wonderful. And thank you. Have a good one.
Okay. Bye.
Bye bye.